So, there's this database called ClinVar, and it's essentially, well, it's like a master catalog of human genetic misery.
Yeah, that's a pretty accurate way to put it.
Right. It's maintained by the National Institutes of Health, and it lists over 75,000 known pathogenic genetic variants.
Wow.
Yeah, 75,000 tiny typos in the human genome that cause diseases. And, we're talking everything from devastating neurological decline to severe immune system failure.
And for decades, the assumption has been that because these typos take so many different forms, right?
Yeah.
Some are missing letters, some are extra letters, or some are just a single wrong letter among 3 billion. We basically assumed we'd need thousands of different custom tools to fix them all.
Exactly. But the premise of our exploration today fundamentally shatters that assumption, because we are looking at a single molecular technology that is theoretically capable of correcting up to 89% of those 75,000 mutations.
It's just staggering when you actually look at the numbers.
It really is. So, welcome to the deep dive. We are taking a massive stack of sources today for you to explore a true paradigm shift in biology.
Yeah, we've got scientific papers from Nature and MDPI, corporate presentations and clinical trial data from Prime Medicine, and, comprehensive reviews from Frontiers and Frontline Genomics.
All of this to decode a revolutionary genetic technology called prime editing.
It's a profound shift in our relationship with the genome, honestly. Like, we are moving away from just treating symptoms or even away from crude genetic disruption.
Right, into this regime of highly precise, intentional rewriting of human DNA. Okay, let's unpack this.
That's good.
Because, you've probably heard the term CRISPR for years, but the tools we're unpacking today make traditional CRISPR look ancient by comparison.
Oh, absolutely, like Stone Age tools versus a scalpel.
Yeah. We are moving from unpredictable genetic scissors to something much more refined, like a molecular machine capable of searching and replacing precise sequences without actually breaking the DNA strands apart.
And that's the key right there.
But before we can understand how prime editing writes new genetic code, we really have to understand the messy, double-stranded reality of the tools that came before it.
Because the story of gene editing is, well, it's really a story of trying to manage cellular tria.
Yeah, that is the perfect context for all of this. To appreciate a surgical tool, you have to understand the damage caused by a blunt instrument.
Right. And for the last decade, the reigning champion of gene editing has been traditional CRISPR-Cas9. And let's establish right away, CRISPR-Cas9 is a monumental discovery.
Oh, for sure. It won the Nobel Prize.
Exactly. It changed biological research overnight. But its fundamental mechanism of action relies on a catastrophic cellular event.
Because it works by creating double-strand breaks in the DNA.
Right. It acts as molecular scissors, just cutting straight through both the Watson and the Crick strands of the DNA double helix at a targeted location.
And our sources emphasize that making that cut is almost trivial for Cas9. The enzyme is incredibly efficient at finding a target and slicing through it.
Yeah, finding and cutting is the easy part.
Right. The actual problem, the chaos, begins the millisecond after that cut is made.
Because a double-strand break is interpreted by a cell as a fatal injury. If a chromosome is broken in half, the genetic instructions are severed.
The cell just panics.
Utter panic. The cell immediately halts its normal cycle, and a wave of panic responses cascade through the nucleus. Proteins like p53 are activated to assess the damage.
And if the break isn't fixed rapidly, the cell will just undergo apoptosis, right? Programmed cell death.
Exactly. To avoid dying, the cell deploys emergency repair mechanisms. And the most common, the immediate response, is a pathway called non-homologous end joining, or NHEJ.
Right. And NHEJ is essentially biological duct tape. The cell isn't looking for accuracy at that point.
No, not at all. It's looking for survival.
It just grabs the two severed ends of the DNA ladder and jams them back together as fast as possible.
Which is a highly error-prone process. The enzymes involved in NHEJ will often chew away a few DNA letters at the broken ends to create a clean surface to glue together.
Or they might haphazardly add a few random letters to bridge the gap.
Yeah, and we call these resulting mutations indels, which is short for insertions or deletions.
This means traditional CRISPR is fantastic if your only goal is to break a gene. Like, if a patient has a gene that is producing a toxic protein, you send in CRISPR, cut the gene.
Let the cell's panicked NHEJ repair system introduce a bunch of random indels.
Right, and the resulting genetic sequence is so garbled that the gene is effectively turned off.
But that gene knockout strategy, Well, it's useful, but it represents a tiny fraction of what we actually need to do in medicine.
Yeah, because the vast majority of those 75,000 variants in the ClinVar database don't need to be broken, they need to be fixed.
Exactly. They are missing a vital function because of a single wrong letter.
And if you want to correct a mutation using traditional CRISPR, you have to rely on a secondary, much less common cellular repair pathway, homology-directed repair, or HDR.
Right. And the limitations of HDR are directly what led to the invention of prime editing.
So, with HDR, you make the double-strand break, but you also flood the cell with a donor DNA template.
Yeah, a synthetic piece of DNA that contains the correct, healthy sequence.
The hope is that the cell will use this donor template as a blueprint to seamlessly stitch the broken chromosome back together, incorporating the healthy sequence in the process.
The intention is sound, but the physics and the biology just fight against you. HDR is highly inefficient.
Why is it so inefficient, practically speaking?
Well, first, it's primarily active only when a cell is actively dividing, in the S and G2 phases of the cell cycle. For postmitotic cells, cells that do not divide, like the neurons in your brain or the cardiomyocytes in your heart, HDR, is virtually non-existent.
Oh, wow. So you simply cannot use it to fix mutations in those tissues at all?
You really can't. And second, even in dividing cells, the messy NHEJ pathway is dominant. It outcompetes HDR almost every time.
So you end up with a mix. A few cells might perfectly incorporate the donor template, but the vast majority just glue the break back together with random errors.
Yeah. And the sources detail an even more severe consequence of making double-strand breaks: chromosomal rearrangements.
Oh, right. This is the nightmare scenario for clinical gene therapy.
Absolutely terrifying. If you introduce CRISPR into a cell and it accidentally cuts at two different places in the genome at the same time, perhaps at the target site and at a similar off-target site, you now have 4 loose ends of DNA floating in the nucleus.
And the cellular repair machinery might grab the wrong ends and fuse them together.
Exactly. You can literally swap large chunks of completely different chromosomes. This is known as a translocation, and it's a known driver of severe cancers, like leukemia.
So the risk of unleashing multiple double-strand breaks in a human patient pushed scientists to look for a way to edit DNA without severing the latter, which brought us to the next generation of tools: base editors.
Right. Base editors were a huge conceptual leap. They proved that you could manipulate the genetic code through direct chemistry rather than, structural violence.
A base editor uses a deactivated or dead Cas9 enzyme, right?
Yes. It uses a Cas9 solely as a navigation system to park at a specific target site. Attached to that parked Cas9 is a chemical enzyme, usually acididine deaminase or an adenosine deaminase.
So instead of cutting the DNA, this enzyme reaches out and chemically modifies the atomic structure of a single DNA letter.
It's fascinating. It strips an amino group off a cytosine molecule, which ultimately tricks the cell into reading it as a thymine. It literally converts a C into a T or an A into a G.
It is elegant chemistry, and it avoids the toxicity of double-strand breaks entirely. But when you look closely at the literature, base editing reveals some pretty severe operational boundaries.
Yeah, very restrictive boundaries. The most obvious is its limited vocabulary.
DNA is made of 4 bases: adenine, cytosine, guanine, and thymine. To fully edit a genome, you need to be able to make 12 possible changes. You need to be able to change any letter into any other letter.
But base editors are strictly limited to what are called transition mutations. They can swap a purine for a purine or a pyrimidine for a pyrimidine. So C to T, G to A to G, T to C.
That's only 4 out of the 12 possible swaps.
Right. The remaining 8 possible swaps are called transversions. Changing a pyrimidine to a purine, like a T to an A or a C to a G.
And traditional base editors cannot execute transversions?
No, their specific chemical deamination process physically cannot force those structural changes. So if a patient's genetic disease is caused by a transversion mutation, a base editor is totally useless to them.
And the sources point out 2 other major flaws. The first is bystander edits.
Oh, yes, the bystander effect.
A base editor operates within a specific editing window, usually a stretch of about 4 to 5 nucleotides on the DNA strand. If you want to change a specific A to a G, but there happen to be 3 other A's sitting right next to it in that window, the DMA's enzyme doesn't discriminate.
It might just convert all 4 A's into G's. You've fixed the pathogenic mutation, but you've created 3 brand new, unknown mutations directly adjacent to it.
The precision is compromised by the immediate local sequence, and then the second flaw is the PAM desert problem.
Right. Traditional Cas9 requires a very specific sequence of DNA, protospacer adjacent motif, or PAM site, to physically latch onto before it can do anything.
For standard Cas9, the sequence is NGG, meaning any nucleotide followed by two guanines. If the mutation you want to fix doesn't have an NG sequence located precisely the right distance away, the base editor cannot dock.
It's like having a helicopter but no landing pad. This severely restricts the number of sites in the human genome that are actually targetable.
So, synthesizing this landscape, we have traditional CRISPR, which is universally programmable but relies on toxic, chaotic double-strand breaks. We have base editors, which are safe and avoid breaks, but have a severely restricted vocabulary and limited targeting range.
Which sets the stage perfectly for late 2019. A postdoctoral fellow named Andrew Ancelone, working in David Liu's lab at the Broad Institute, publishes a paper introducing a system that bypasses almost all of these limitations.
Prime editing.
The true search and replace word processor. It was a watershed moment in molecular biology. Anzalone and Liu designed a system that does not require double-strand breaks. It doesn't require separate exogenous donor DNA templates to be flooded into the cell.
And it can perform all 12 possible base swaps, both the transitions and the transversions.
Yes. Furthermore, it can perform highly targeted, precise insertions of new DNA sequences and targeted deletions of pathological sequences.
This brings us back to that ClinVar statistic I opened with. Because prime editing isn't restricted by the chemistry of deamination, and because it can handle those small insertions and deletions, it covers the vast majority of the genetic error landscape.
That 89% coverage is theoretical, assuming optimal delivery and targeting, of course, but the mechanical capability is absolutely there.
It unifies the safety profile of base editors with the broad utility of homology-directed repair,
Entirely circumventing the destructive elements of both.
I think we need a solid analogy to visualize this progression. If traditional CRISPR is a machete hacking through a sentence to splice in a new word,
Good analogy, yeah.
and base editing is like a limited autocorrect that might accidentally change bat to cat when you really want a Bart,
Right.
then prime editing is a precise word processor where you simply hit find and replace.
The evolution from destruction to chemical conversion to outright programmatic rewriting?
But wait, if we are calling it a word processor, there is a fundamental mechanical question we have to answer here.
Okay, what's that?
If prime editing is not cutting the DNA ladder in half, how on earth is it physically inserting new genetic information into a sealed double helix? The ladder is closed, the bases are paired up. How does new information get inside?
That is the central biomechanical paradox that the Liu lab had to solve. To understand how prime editing sneaks new sequence data into the genome without shattering the chromosome, we have to examine the heavily engineered hardware and software components of the system.
It involves hijacking cellular mechanics in a very specific way, right? Let's look at the actual parts. Our sources break down the prime editing system into two massive components. The hardware is the prime editor protein, or PE. The software is the prime editing guide RNA, or page RNA.
Let's start with the hardware. It is a fusion protein.
Meaning, scientists have taken two distinct molecular machines that evolved for completely different purposes and physically tethered them together into a single continuous chain of amino acids.
Exactly. And the first half of this fusion is, once again, a Cas9 enzyme, but it has been surgically modified.
The sources refer to it as a Cas9 nickase, specifically carrying the H840A mutation. Let's break down exactly what that means.
Sure. So standard Cas9 has two distinct molecular blades cutting domains. One is called the H&H domain, which cuts the target strand of the DNA. The other is the RuVC domain, which cuts the non-target strand.
And working together, they create the double-strand break.
Right. In the prime editor hardware, researchers introduced a tiny tweak. They changed the 840th amino acid in the Cas9 sequence from a histidine to an alanine. This mutation, H840A, completely inactivates the H&H cutting domain.
It effectively dulls one of the blades.
Precisely. The Cas9 nickase retains its ability to navigate the genome and pry the two strands of DNA apart, but when it engages its cutting mechanism, only the RuVC domain is active. It only slices the non-target strand.
So the backbone of the chromosome remains intact on the opposite side. It just creates a little flap, a small opening on one side of the ladder.
Yeah, just a tiny nick. Now, attached to the back end of this modified Cas9 is the second half of the hardware, a reverse transcriptase enzyme, or RT.
And the specific RT used here is derived from the Maloney marine leukemia virus, or MMLV.
This is a profound instance of biological repurposing. Reverse transcriptases are engines of viral replication. Retroviruses like the murine leukemia virus, or HIV, carry their genetic material as RNA.
When they infect a host cell, they use reverse transcriptase to read their viral RNA and translate it backward into DNA, which is then integrated into the host's genome.
Yes, Anzalone and his team isolated this viral ability to write DNA from an RNA blueprint and directly attach to the Cas9 nickase.
It's an incredible hybrid. I mean, you have a bacterial immune system, which is what Cas9 originally evolved as, right, to chop up invading bacteriophages, and you have fused it to a viral replication engine.
You are using the tools of infection and immunity as a localized construction crew. The Cas9 provides the precise localization and opens the workspace, while the viral RT provides the synthetic capability to write the new code.
But the hardware is blind. The Cas9 nickase and the reverse transcriptase have no idea where to go or what sequence to write.
That brings us to the software. The PEG-RNA, the Prime Editing Guide RNA. This is a masterpiece of RNA engineering.
Because traditional CRISPR uses a simple guide RNA that serves only as a GPS coordinate. It finds the sequence and parks.
But the PEG-RNA has to do much more. It has to act as the GPS, the scaffolding for the protein complex, the anchor for the cut DNA, and the actual blueprint containing the new genetic sequence.
Let's walk through the physical structure of the PEG-RNA. It is built in 4 contiguous sections. The first is the spacer.
The spacer is the navigation unit. It is a sequence of typically 20 nucleotides that continuously scans the billions of base pairs in the human genome until it finds its exact complementary match at the target site.
Once it finds the site, we rely on the second section, the scaffold.
The scaffold is the structural backbone. It forms complex three-dimensional loops of RNA that the Cas9 protein physically grips onto. It ensures the hardware and software stay permanently tethered together as they move through the nucleus.
Now the architecture gets entirely novel for prime editing. The third part of the PEG-RNA is the primer binding site, or PBS.
This is the first crucial innovation that allows prime editing to bypass double-strand breaks. Remember that the Cas9 nickase just made a tiny single-stranded slice on the non-target DNA strand? That nick creates a loose, dangling end of DNA. The primer binding site is a sequence of RNA designed to perfectly complement that newly exposed loose DNA end.
Wait, so it's acting like a molecular tether? It reaches out, grabs that loose piece of nicked DNA, and binds to it?
Exactly. It hybridizes with it. By binding to that loose DNA end, the PBS physically anchors the genomic DNA directly to the PEG-RNA molecule. It stabilizes the open workspace.
Which sets up the fourth and final part of the PEG-RNA, the reverse transcription template, or RTT.
The RTT is the payload. It is the sequence of RNA that physically holds the new, corrected genetic information.
So once the PBS has anchored that loose piece of nicked DNA, the viral reverse transcriptase enzyme recognizes this specific junction.
It docks at the site where the DNA and RNA are hybridized. The enzyme looks at the RTT RNA sequence and it begins to synthesize new physical DNA directly onto the loose end of the nicked strand, extending it letter by letter.
This directly answers how the new letters get inside the closed double helix. The system uses the cell's own mechanical tension against it. It doesn't shatter the latter. It just nicks one side, peels back a tiny flap of the original DNA, tethers that flap to an RNA blueprint, and literally prints the new letters directly onto the open rome.
It creates a localized transient opening, performs the localized synthesis, and prepares to seal it back up. What's fascinating here is how the system hijacks the cell's own mechanics to safely open the DNA just enough to slide in a new sequence.
Describing the individual components is helpful, but to truly grasp the thermodynamics and the cellular reality of this, we need to view it in motion. Let's run a chronological play-by-play of the exact sequence of events when this massive molecular machine lands on a target inside a living cell.
Let's break down the molecular choreography. Step 1 is navigation and unzipping. The prime editor complex, guided by the PEG-RNA, locates the target sequence. The Cas9 protein forces a double helix to unwind locally, creating a small open bubble of single-stranded DNA.
Step 2, the nick. The inactivated H&H domain does nothing, but the active RuVC domain slices the non-target strand, precisely 3 base pairs upstream of the PAM sequence.
Step 3, flap generation. Because of that precise nick, the DNA strand is severed. This creates a loose, single-stranded end. In molecular biology, this loose piece of DNA is technically referred to as a flap.
Step 4, hybridization. The primer binding site on the PEG-RNA reaches out and hybridizes to the 3' end of this newly created DNA flap.
And the orientation here is critical. DNA has directionality, defined as 5' to 3'. DNA synthesis can only occur by adding new nucleotides to a 3' end. By hybridizing to the 3' flap, the pgRNA provides the necessary starting block for the next step.
Step 5. Synthesis. The reverse transcriptase enzyme engages the bound 3' flap. It reads the adjacent RTT sequence on the pgRNA and begins polymerizing new DNA, extending the flap with the corrected sequence.
Now, at the end of synthesis, we arrive at the most chaotic and fascinating moment of the entire process. The RT has just synthesized a brand new stretch of DNA, but the original unedited DNA hasn't gone anywhere.
It's still physically attached to the chromosome, directly adjacent to our new edit.
Right. We have a physical traffic jam on the double helix. There are now two overlapping pieces of single-stranded DNA competing to bind to the exact same spot on the opposite unedited strand.
We have the newly synthesized 3' flap, which contains our corrected sequence, and we have the original unedited 5' flap, which contains the disease mutation we want to displace. They cannot both occupy the same space.
The sources describe this state as equilibration. The two flaps are dynamically shifting, binding and unbinding, fighting for hydrogen bonds with the opposite strand.
And if the old 5' flap wins and binds back into place, our new edit is pushed out, forming a loose tail that will eventually be degraded by the cell. The edit fails.
But the architecture of prime editing relies on a thermodynamic advantage and the presence of a specific cellular janitor. Human cells are constantly dealing with overlapping flaps of DNA naturally, especially during normal DNA replication.
Right, when creating Okazaki fragments on the lagging strand.
Exactly. Because of this, the nucleus is filled with cellular endonucleases, whose sole job is to clean up these chaotic flap structures.
And the specific hero of this story is an endonuclease called FEN1, flap endonuclease 1.
FEN1 has a very specific biochemical preference. It is designed to recognize and cleave 5' flaps. It generally ignores 3' flaps.
So as these two strands are competing, FEN1 is patrolling the nucleus.
It encounters this complex overlapping structure, recognizes the old unedited 5' flap, and chemically snips it off.
Wow, so the cell's own internal garbage disposal system actively removes the diseased gene sequence for us.
It does. Once FEN1 excises the old flap, the new edited 3' flap drops cleanly into place, pairs with the opposite strand, and naturally occurring DNA ligase seals the nick, locking the sugar phosphate backbone back together.
It is a remarkable cascade. You introduce the viral RT to write the code, but you rely entirely on the cell's endogenous replication cleanup crew to remove the original mutation and finalize the physical integration.
Yeah.
But, okay, tracing this logic creates an immediate glaring structural problem.
What's that?
We just replaced one side of the DNA ladder, we wrote the new letters, FEN1 chopped away the old letters, and the ligase sealed the new side in. But DNA has two sides. We only edited the non-target strand.
Right.
If the original mutation was a TA base pair and we just changed the T to a C, well, the bottom strand is still an A. A and C do not bind together. Doesn't this create a mismatch where the two strands don't pair up properly?
You have a physical distortion in the double helix. The hydrogen bonds cannot form. You have created a heteroduplex, severe mismatch.
Doesn't the cell's natural repair system just look at this mismatch, freak out, recognize the structural bulge, and potentially fix our new edit right back to the disease state?
You are exactly right. You have pinpointed the single greatest bottleneck in the development of this technology. The cell's mismatch repair system, or MMR, is a highly aggressive proofreading mechanism.
Complexes of proteins called MUT-S and MUT-TL constantly scan the genome for these exact types of bulges, right?
Yeah. When they find a mismatch, they rip out the surrounding DNA and resynthesize it. If the MMR system arbitrarily chooses the unedited strand as the correct template, it will completely erase the prime edit.
This tug-of-war with the mismatch repair system is why the technology didn't stop at Andrew Anselon's first prototype in 2019. The NDPI and Frontiers papers detail an aggressive, iterative engineering effort. Prime editing isn't a single static protein, it's a continuously updated software suite. We have PE1, PE2, PE3, PMAX, VPE.
We have to examine how scientists essentially waged war against the cell's own proofreaders. The progression of prime editor variants is a masterclass in directed evolution and rational protein design.
Let's start with the transition from PE1 to PE2.
PE1 was the initial proof of concept. It utilized a wild-type, totally unmodified reverse transcriptase. It demonstrated that the flap mechanism could work, but it was incredibly inefficient. The wild-type RT simply wasn't designed to operate in the complex, heavily regulated environment of the human nucleus.
So for PE2, the Lu lab focused entirely on upgrading the engine. They introduced 5 distinct targeted mutations into the reverse transcriptase enzyme itself.
These were not random mutations. They were selected to enhance specific physical properties of the enzyme. They increased its thermostability, allowing the RT to maintain its complex folding structure at mammalian body temperatures without denaturing.
And they increased its processivity, meaning its physical grip on the DNA-RNA substrate was strengthened so it could write much longer stretches of new DNA without prematurely falling off the template.
And PE2 was a massive success on that front, increasing editing efficiency up to 5-fold compared to the PE1 prototype.
But PE2 still leaves us stranded with the mismatch problem. The new strand is edited, the bottom strand is the original disease sequence, and the MMR system is closing in to fix it.
This is where biological cunning comes into play. To solve the mismatch problem, the host pointed out, researchers developed the PE3 strategy. The fundamental challenge was, how do you convince the cell's mismatch repair machinery that the newly synthesized edited strand is the original healthy truth, and that the unmutated original strand is the error that needs to be deleted?
You essentially have to frame the healthy strand for a crime.
That is an excellent way to conceptualize it. The MMR system has a built-in heuristic. It assumed that if one strand of DNA contains a recent nick or break, that strand must be the damaged, newly synthesized, or incorrect strand. It uses the intact strand as the reliable template.
So the PE3 system introduces molecular sabotage. Along the prime editor complex in the primary PEG RNA, you introduce a second standard guide RNA.
This second guide doesn't carry any new edits. It just directs the Cas9 nickase to make a single, simple cut on the unedited opposite strand of the DNA, usually a few dozen base pairs away from the primary edit.
So the cell's mismatch repair system detects the mismatched base pairs caused by the prime edit. The MUD-S complex binds to the bulge, but then it scans the surrounding DNA and discovers that the unedited strand has a fresh, bleeding nick in it.
Obeying its biological programming, the MMR system concludes that the nicked, unedited strand is the damaged error. It physically degrades that unedited strand, and it uses our newly edited strand as the template to rebuild the opposite side.
It naturally copies our edit over to the other side of the ladder. It locks the change into both strands, making it a permanent homozygous edit.
The sheer ingenuity of hijacking a repair mechanism by just lightly nicking the opposite side is staggering.
It is brilliant, but it is a delicate balance, right? Nicking the opposite strand carries a risk.
Yeah, big risk. If the nick on the edited strand and the nick on the unedited strand happen too close together in time and physical space, you accidentally recreate a double-strand break, which is exactly what prime editing was designed to avoid. The timing must be perfect.
And the sources note that even with the PE3 nicking strategy, the mismatch repair system is sometimes just too fast and too aggressive. In certain cell lines, MMR would jump in and erase the primary edit before the secondary nick could even be established. The efficiency plateaued.
Which necessitated the development of PE4 and PE5. If you cannot trick the security guards fast enough, the alternative is to temporarily drug them.
The PE4 and PE5 systems employ a strategy of transient suppression. Along with the prime editor, they co-express a protein called MLH1DN. Let's break down MLH1DN. MLH1 is a core protein in the MUT-TL complex of the mismatch repair system.
Yeah.
What does the DN stand for?
It stands for dominant negative. A dominant negative protein is a mutated version of a natural protein that can still bind to its target but cannot perform its function. In this case, MLH1DN binds to the mismatch repair machinery and physically jams it.
It essentially acts as a chemical pause button for the cell's proofreading system.
Exactly. So we deploy the prime editor and simultaneously flood the nucleus with this dominant negative protein to shut down the cell's error-checking algorithms.
We make the edit, allow the 3' flap to settle in, allow the secondary nick to lock it in, and then as the MLH1DN protein naturally degrades over a few days, the mismatch repair system wakes back up.
And by the time the MMR system comes back online, the edit is seamlessly integrated into both strands of the DNA. There are no mismatches left to find. The deployment of PE4 and PE5 massively boosted successful edit retention across a huge variety of human cell types.
The level of iterative optimization is relentless. The literature also covers PMAX, which focuses on the nuclear localization signals, the molecular zip codes that tell the cell to import the massive prime editor protein into the nucleus where the DNA actually lives. By optimizing these signals and tweaking the human codon usage, they ensured higher concentrations of the editor actually reach the workspace.
And we must discuss the VPE variant, the validated prime editor. We talked earlier about the equilibration of the flaps. While FEN1 usually cleans up the 5' flap perfectly, It is not an infallible process.
Right.
Occasionally, as the 3' flap drops into place, the local sequence might cause it to fold back on itself or misalign by a single base before the ligase seals it.
The sources refer to this as misaligned flap resolution, leading to tiny unintended insertions or deletions at the edit site.
The VPE variant addresses this directly. By introducing highly specific structural mutations into the reverse transcriptase and the Cas9 interface, VPE alters the physical tension during the flap equilibration phase.
These structural tweaks hold the DNA strands in a more rigid conformation, drastically reducing the chances of the flap misaligning.
The data shows VPE reduces these specific error rates by up to 60-fold compared to previous versions. If we connect this to the bigger picture, this shows how genome editing has shifted from pure discovery to iterative, software-like updates.
This completely reframes how we should think about genetic medicine. We aren't just discovering enzymes in nature and hoping they work. We are executing software patches. We are at version 5.0, tweaking lines of biological code to increase processivity, adding transient inhibitors to bypass specific error-checking algorithms. We are treating human cellular biology as an operating system.
It is the pinnacle of rational bioengineering. The fundamental architecture was conceptualized in 2019, and the subsequent years have been a global, collaborative effort of iterative debugging.
But all this elegant biochemistry, all these thermodynamic flap battles, they are completely meaningless unless they actually cure human beings. We can marvel at reverse transcriptase all day, but the ultimate goal is to alleviate human suffering. Let's move from the theoretical physics of the nucleus to the actual clinical data. Let's look at the historic first-in-human trials.
This is the moment where the abstract becomes profoundly tangible. As of late 2025 and into 2026, Prime Medicine has successfully navigated the technology out of the petri dish and into human patients. Their flagship clinical program is the PM359 trial.
We have the specific clinicaltrials.gov record right here in our sources. NCT 06559176. This is a phase 12 trial for a disease with a very intimidating name. P47, Fox autosomal recessive chronic granulomatous disease, we'll just call it CGD. What is the physical reality of this disease for a patient?
CGD is a rare, devastating inherited immunodeficiency. The disease stems from a mutation in a gene called NCF1. This gene is responsible for producing a crucial subunit of an enzyme complex called NADPH oxidase, which is utilized by a specific type of white blood cell known as the neutrophil.
And neutrophils are the first responders of the immune system. They patrol the bloodstream, and when they encounter a bacterial or fungal pathogen, they physically engulf it in a process called phagocytosis. They eat the invader.
Yes, but eating the bacteria isn't enough. The neutrophil has to kill it. To do that, the NADPH oxidase complex generates a massive burst of reactive oxygen species, essentially molecular bleach inside the neutrophil to destroy the engulfed pathogen.
But in CGD patients, because of the NCF1 mutation, the NADPH oxidase complex is broken. The neutrophils can eat the bacteria, but they cannot produce the reactive oxygen to kill it.
The bacteria just survives inside the immune cell. The immune system, unable to clear the infection, simply walls off the area, creating massive inflammatory masses called granulomas. These patients suffer from severe, recurrent, life-threatening bacterial and fungal infections.
A simple fungal spore from garden soil, which your immune system clears without you ever knowing, can cause a lethal pneumonia in a CGD patient.
And the specific genetic error driving this entire systemic collapse is often shockingly small. The sources note it is frequently a del-GT mutation.
Just two specific letters, a guanine and a thymine, are deleted from the DNA sequence. 2 missing letters out of 3 billion, and the immune system fails.
That small deletion shifts the reading frame of the gene, resulting in a truncated, useless protein.
So how does the PM359 prime editing therapy actually address this? The clinical trial outlines a highly choreographed ex vivo procedure.
Ex vivo means the actual genetic editing occurs outside the patient's body. The first step involves treating the patient with specific mobilizing drugs like G-CSF or plerixafor. These drugs force the bone marrow to release its precious stem cells directly into the peripheral bloodstream.
The patient then undergoes apheresis, a process where their blood is continuously routed through a machine that centrifuges and filters out a very specific population of cells. CD34 plus hematopoietic stem cells. I want to clarify what CD34 plus means because it's crucial.
Oh, definitely.
CD34 is a protein acting as a surface marker. It identifies the cell as a hematopoietic stem cell, the grandfather cell of the immune system. This single cell has the capability to differentiate and continuously produce millions of new white blood cells, red blood cells, and platelets for the rest of the patient's life.
If you edit a mature neutrophil, it dies in a few days and the disease remains.
Hmm.
If you edit the CD34 plus stem cell, you edit the entire downstream lineage permanently. You are editing the factory, not the product.
These collected CD34 plus stem cells are shipped to a highly regulated central manufacturing facility. Inside the lab, the cells are temporarily permeabilized, and the PM359 prime editor components, the engineered protein, and the specific pegRNA are introduced into the cells.
The editor navigates to the NCF1 gene, locates the exact site of the del-GT mutation, and utilizes the reverse transcriptase mechanism we discussed to literally print the missing G and T directly back into the sequence.
It restores the reading frame perfectly without ever severing the chromosome. Now, the lab possesses a batch of fully corrected stem cells capable of producing fully functional, pathogen-killing neutrophils. But the clinical process isn't as simple as just putting them back in immediately, is it?
No, because the patient's bone marrow, the physical space inside their bones, is still entirely occupied by their original diseased stem cells. The new cells need a niche to engraft and multiply, so the patient must undergo a preparative regimen known as conditioning.
This typically involves the administration of a potent chemotherapy agent like busulfan. Busulfan systematically destroys the existing bone marrow. It is an intense, physically grueling process for the patient, temporarily leaving them with zero immune protection.
Once the bone marrow niche is cleared, the perfectly edited PM359 stem cells are infused back into the patient via standard intravenous drip. The stem cells naturally home back to the bone marrow compartments, they engraft into the emptied niches, and they begin the process of hematopoiesis, producing a lifelong, self-renewing supply of healthy immune cells.
The mechanism is awe-inspiring, but the clinical reality is what matters. We have a press release from December 2025 detailing the status of the first 2 patients treated in the PM359 trial.
The initial data is historic. The release states that the first 2 patients have been effectively cured of CGD.
Effectively cured?
Yes. Following engraftment, their bodies are producing neutrophils with functional NADPH oxidase complexes. Their immune systems can now generate the reactive oxygen bursts necessary to kill pathogens. The biological defect has been permanently erased.
It is a profound milestone in medical science. But Prime Medicine's corporate presentations outline a pipeline that extends far beyond CGD. They are aggressively targeting a diverse array of mutations to showcase the versatility of the Prime Editor platform. For instance, they are developing a treatment for sickle cell disease.
Sickle cell disease is caused by a very specific transversion mutation in the HBB gene. A single adenine is mutated into a thymine. This changes the structure of hemoglobin, causing red blood cells to deform into a sickle shape, leading to excruciating pain crises and organ damage.
Remember, traditional base editors cannot fix a T2A transversion, but prime editing can swap the rogue thymine right back to an adenine, reverting the gene to the wild-type healthy sequence.
The presentations also detail a program for Tay-Sachs disease. Tay-Sachs is a devastating, fatal neurological disorder often caused by a 4-base pair insertion in the HEKXA gene. 4 extra letters cause the accumulation of toxic lipids in the brain.
Prime editing is programmed to simply target that specific 4-base sequence and delete it, restoring the precise original code.
Look at the sheer mechanical diversity of those 3 targets. CGD required the insertion of 2 missing letters. Sickle cell required the targeted transversion swap of a single letter. Tay-Sachs required the deletion of 4 extra letters.
A single technological platform utilizing the exact same Cas9-RT fusion protein accomplished all 3 distinct tasks simply by changing the RNA sequence in the pegRNA software.
It's remarkable.
But there is another program detailed in the corporate presentation that represents an even larger technological hurdle, PM577, their therapy for Wilson disease. Because unlike CGD or sickle cell, where you can extract the stem cells, edit them safely in a lab, and infuse them back, Wilson disease requires an in vivo approach. You have to edit the cells while they are still inside the living patient's organ.
Wilson disease is a severe metabolic disorder. The body loses the ability to properly excrete copper. The primary site of this failure is the liver. The copper accumulates to highly toxic levels in the liver tissues, eventually spilling over into the bloodstream and accumulating in the brain, leading to hepatic failure and severe neurological deterioration.
It is frequently caused by a specific point mutation known as H1069Q in the ATP7B gene.
Right, you cannot extract a patient's liver put it in a centrifuge, edit the cells, and put it back. You have to send the massive prime editor complex directly into the human body and ensure it reaches the correct tissue.
With CGD, they take the cells out of the body to edit them. With Wilson disease, they are injecting lipid nanoparticles directly into the liver. Why use different methods, and what does this say about the versatility of the technology?
Well, they bypass viral vectors entirely for this liver indication. PM577 packages the genetic instructions for the prime editor, specifically the mRNA encoding the PE protein and the synthetic PEG RNA inside lipid nanoparticles, or LNPs.
LNPs gain global recognition as a delivery vehicle for the mRNA COVID-19 vaccines. They are essentially microscopic spheres of engineered synthetic fat.
Precisely. The LNP encapsulates the fragile RNA instructions, protecting them from degradation in the bloodstream. The patient receives an intravenous infusion of these LNPs. Because of the specific biochemical properties of the lipids used and the sheer volume of blood flow through the organ, these LNPs naturally accumulate in the liver.
They fuse with the lipid membrane of the hepatocytes, the functional cells of the liver, and release the prime editor mRNA directly into the cellular cytoplasm.
The hepatocyte then translates the mRNA into the physical prime editor protein, which enters the nucleus and corrects the ATP7B gene.
The data presented from the fully humanized mouse models for PM577 is remarkable. The mice carry the human Wilson disease mutation. After a single intravenous LNP treatment, the researchers observed greater than 80% correction of the target gene across the entire population of liver hepatocytes.
An 80% correction rate in vivo is an astronomical success. In the context of metabolic liver diseases, correcting even 10 to 15% of the hepatocytes is often sufficient to restore enough enzyme function to eliminate the disease symptoms. Reaching 80% implies that the liver has been fundamentally holistically restored to wild-type function.
The mice demonstrated entirely normal hepatic copper concentrations within 8 weeks, safely excreting the copper through biliary pathways. Taking a step back and comparing CGD to Wilson disease highlights the absolute modularity of this technology platform.
You choose the delivery vehicle based entirely on the tissue. Blood diseases rely on ex vivo stem cell apheresis. Solid organ diseases like the liver rely on in vivo lipid nanoparticles. But the core editing engine, the thermodynamics of the flap, the reverse transcriptase, the guide RNA logic, remains exactly the same. You just change the delivery track.
We are transitioning away from chronic disease management. We are moving from a paradigm of taking drugs every day for the rest of your life to manage toxic copper levels or living on prophylactic antibiotics to survive an immunodeficiency to executing a single permanent genetic reversal at the foundational sequence level.
However, the clinical applications for human disease, as monumental as they are not the only areas where prime editing is establishing a foothold.
While curing human genetic disease is the headline, prime editing is quietly revolutionizing other fields of biology, from agriculture to the creation of bespoke diseased cells for research.
This specific application is arguably just as impactful for the broader scope of global health, even if it doesn't generate the same clinical headlines. We are talking about highly advanced disease modeling.
The MDPI paper details how scientists are using prime editing not to cure diseases, but to intentionally cause them in healthy cells, which initially sounds entirely counterintuitive. Why go through the effort of building this massive molecular machine just to break a healthy human cell?
Because if you want to study the intricate progression of a rare genetic disease, or if you want to perform high-throughput screening to find a small molecule drug that treats it, you need millions of human cells that actually carry that exact pathogenic mutation. Historically, obtaining those cells was a massive bottleneck. You had to find a human patient with that specific rare variant, perform a tissue biopsy, and attempt to culture their fragile cells in a lab.
But with prime editing, you don't need the patient's tissue. Researchers can take readily available, healthy, human, pluripotent stem cells. They can use chemical signals to coax these stem cells to grow into miniature, three-dimensional rudimentary organs in a petri dish, known as organoids. And then they deploy prime editing to seamlessly install the exact pathogenic mutation they want to study.
By perfectly recreating the disease genotype in a highly controlled, sterile environment, they create an infinite supply of test subjects. The MDPI paper lists several prominent examples. Researchers successfully installed the specific CFTR mutation responsible for cystic fibrosis directly into intestinal organoids to study the exact mechanisms of mucus buildup.
They introduced the ABCB11 mutation into liver organoids to study the cellular mechanics of severe bile salt export pump deficiency.
They even mentioned utilizing prime editing in animal models for oncology. They precisely placed the KRAS mutation, which is a notorious driver of pancreatic and lung cancers, directly into the genome of zebrafish.
Because zebrafish are largely transparent during early development, researchers can literally watch the tumor initiate and develop in real time under a microscope, driven by the exact human mutation.
It accelerates fundamental biological discovery. By utilizing the precision of prime editing, scientists know with absolute certainty that any cellular dysfunction they observe is caused only by the single mutation they installed, without the background noise of off-target double-strand breaks clouding the data.
And the sources push even further, detailing the massive implications for agriculture. Prime editing is being deployed to alter the genetics of the global food supply.
The agricultural sector faces immense pressure. Climate change is altering weather patterns, introducing new pathogens, and causing severe drought. We need crops that are resilient, but traditional selective breeding takes decades. Traditional genetic modification, while faster, comes with immense regulatory and societal baggage.
The sources specifically mention the use of prime editing to transfer single nucleotide polymorphisms, or SNPs, between different crop varieties. They highlight successes in rice, peanuts, and chickpea genomes. Let's explore why this is legally and socially so distinct from what the public knows as GMOs.
Traditional genetic modification in agriculture often involves transgenic techniques. This means introducing entirely foreign DNA into a plant's genome. For example, taking a gene from a soil bacterium that produces a natural insecticide and inserting it into the genome of a corn plant. This creates a transgenic organism. It is highly effective, but it triggers intense regulatory scrutiny and significant public pushback due to the perception of creating frankenfoods.
But prime editing does not require the insertion of exogenous foreign DNA. You aren't taking bacterial genes and jamming them into a peanut. You are utilizing the prime editor complex to simply rewrite the peanut's own endogenous DNA. You might change a single letter in a water retention gene to perfectly match a naturally occurring, highly drought-resistant, but lower-yield strain of that exact same peanut species.
Exactly, because prime editing relies entirely on the temporary RNA template and does not create shattering double-strand breaks. The resulting genomic edit is biochemically indistinguishable from a natural evolutionary mutation that could have occurred over thousands of years of selective breeding. It is simply accelerated evolution.
This precision potentially bypasses the heavy transgenic regulatory hurdles entirely. You can engineer crops to be climate resilient, disease resistant, or nutritionally fortified in a fraction of the time, utilizing only the genetic vocabulary native to that species.
It is a vital tool for ensuring global food security in an increasingly unstable climate.
It sounds like magic, but the sources explicitly warn us of severe, ongoing bottlenecks. It's time to look at the glaring limitations of the technology.
A rigorous scientific assessment demands it. For all its thermodynamic elegance on a localized molecular level, translating prime editing into a widespread, easily deployable therapeutic reality is fraught with immense engineering challenges. And the single most significant bottleneck right now is the delivery problem.
It is an issue of physical mass. The prime editor complex is massive. The sources list the molecular weight. The combined sequence of the Cas9 nickase fused to the Maloney-Marine leukemia virus reverse transcriptase is approximately 6.3 kilobases in genetic size.
To place that number in context, we have to look at how we normally deliver genetic medicine. The safest, most heavily utilized, and FDA-approved viral vector for delivering gene therapy into human cells is the adeno-associated virus, or AAV. It is the absolute workhorse of the industry. It can target a wide variety of tissues safely. But AAVs have a hard physical limitation. The absolute maximum cargo capacity of an AAV shell is about 4.7 kilobases.
You physically cannot fit the 6.3 kilobase prime editor into the standard 4.7 kilobase delivery truck. It just doesn't fit.
Exactly. The payload is too big. This forces researchers to engineer complex, inefficient workarounds. We discussed lipid nanoparticles earlier, which are excellent for the liver. But LNPs struggle significantly to reach other organs efficiently. If you want to edit cells in the brain, the heart, or the muscles, LNPs are currently highly inefficient at crossing those specific biological barriers. You need viral vectors like AAVs to reach those tissues.
So if you must use AAVs, but the editor doesn't fit, how do they solve it? The sources mention splitting the editor in half.
Yes, the dual AAV system. Researchers literally cut the prime editor genetic sequence into two pieces. They package the front half of the protein in one AAV and the back half of the protein in a second AAV. They use a technology called splitentines, which act like molecular Velcro.
How does that actually work inside the patient?
You infuse billions of both types of AAVs into the patient. You have to hope that both virus A and virus B infect the exact same target cell at the exact same time. If they both deliver their half of the sequence into the nucleus, the two halves are translated into proteins, the split inteins find each other, and they spontaneously stitch the two halves back together into a functional prime editor complex.
That sounds incredibly convoluted. The statistical probability of both viruses successfully infecting the exact same cell and synthesizing their halves in sufficient quantities at the same time must drastically reduce the overall editing efficiency.
It does. It is a statistical numbers game, and it significantly lowers the potency of the therapy compared to a single vector delivery. Researchers are exploring alternatives, like packaging the editor into virus-like particles or VLPs. These are engineered hollow protein shells that are much larger than AAVs but lack viral genetic material. However, VLPs face massive manufacturing bottlenecks and have their own distinct tissue targeting challenges. Delivery remains the primary hurdle for systemic in vivo therapies.
But the delivery of the hardware isn't the only challenge. The literature points out that simply designing the software, the pegRNA, is incredibly difficult. The MDPI paper explicitly notes that designing a highly efficient pegRNA is currently more art than science.
The complexity of the pegRNA is a double-edged sword. We discussed its four structural components: the spacer, the scaffold, the primer binding site, and the reverse transcription template. The lengths of the PBS and the RTT are critical variables. If the PBS sequence is just one or two nucleotides too short, it lacks the thermodynamic stability to properly hybridize and anchor the loose DNA flap. If the RTT sequence is too long, the reverse transcriptase might stall out, or the RNA might fold in on itself, forming secondary structures that block the enzyme entirely.
So a researcher cannot simply feed the target sequence into a computer algorithm, hit print, and expect a perfect pegRNA.
Not yet. The algorithms are improving, but currently, optimizing a single edit requires immense empirical testing. Researchers have to synthesize matrices of dozens or hundreds of different pegRNA designs, varying the lengths of the PBS and RTT by single nucleotides, and physically test every single combination in human cell lines to find the one configuration that yields the highest editing efficiency with the lowest error rate for that specific genomic locus. It is an incredibly painstaking, labor-intensive process.
And while prime editing is unprecedented in its ability to handle small edits, swapping single letters, or deleting a handful of bases, the traditional PE system struggles fundamentally with large, multi-kilobase edits.
Yes. The reverse transcriptase enzyme used in the fusion protein is highly processive for short stretches, but is not designed to synthesize thousands of base pairs. If a genetic disease requires the insertion of an entire full-length healthy gene, perhaps a sequence that is 2,000 or 3,000 letters long, the traditional prime editor will physically fall off the DNA template before it finishes copying the massive sequence.
I noticed in the Prime Medicine corporate presentation that they are actively developing newer iterations to bypass this size limit. They highlight technologies called Passage and Twin PE. How do these systems attempt to insert large payloads?
They utilize prime editing to perform a setup maneuver. Instead of trying to write the entire massive gene with the reverse transcriptase, they use prime editing to simply install a small, highly specific sequence into the genome called a landing pad. Once that landing pad is safely integrated into the DNA, they deploy a completely separate class of enzymes known as site-specific recombinases or integrases like the BXB1 integrase.
So prime editing acts as the advanced team preparing the site.
Exactly. The integrase recognizes the newly installed landing pad, and it is capable of taking a massive multi-kilobase exogenous DNA payload and seamlessly plugging it directly into that specific site. It is essentially using prime editing to install a highly secure USB port into the genome, allowing you to plug in a massive file later.
It is a brilliant workaround, but it adds entirely new layers of complexity. Now you have to deliver the prime editor, the guide RNAs, the massive DNA payload, and the integrase enzyme all to the same cell.
The complexity compounds exponentially. And beyond the pure technical hurdles of delivery and payload size, we must confront the clinical and corporate risk. As candidly noted in Prime Medicine's own financial disclosures to their investors, the ultimate reality of any genetic therapy is its permanence.
You are rewriting the foundational hard drive of the human cell. There is no undo button.
If an unintended consequence occurs, if the editor generates an off-target edit that inadvertently mutates a tumor suppressor gene, potentially initiating a cancer down the line, or if a misedit occurs directly at the target site, the therapy cannot be withdrawn. You cannot simply instruct the patient to stop taking a pill. The altered genetic sequence is locked into their stem cells, and every subsequent daughter cell produced for the rest of their life will carry that alteration.
And we discussed earlier that the flap equilibration process is not perfectly flawless. The battle between the 5' and 3' flaps can occasionally result in misaligned flap resolution.
The data in the sources detail this reality clearly. Depending on the specific prime editor system utilized, the specific cell type, and the local thermodynamic landscape of the target sequence, these small unintended misedits can occur at varying rates, ranging anywhere from 1 in 7 edits to 1 in 121 edits. The newer VPE variant has drastically suppressed these rates, but when a therapy involves editing millions or billions of stem cells across a patient's entire body, even a 1 in 1,000 error rate translates to thousands of cells carrying brand new unintended mutations.
It forces a profound regulatory and ethical calculation. This raises an important question regarding the permanence of these therapies. How does an agency like the FDA regulate a cure that writes permanent changes into a living human being, knowing with statistical certainty that there is a non-zero chance of generating a novel, unintended mutation in a fraction of those cells?
It represents a staggering regulatory challenge. The FDA is engaging closely with developers, granting specific accelerated pathways for these rare diseases, but the ultimate long-term safety data simply does not exist yet. It cannot exist. We will not fully comprehend the true lifetime risks of these subtle off-target effects until the current clinical trial patients have been monitored for decades.
So what does this all mean? The host brings it back to the listener, summarizing the duality of prime editing. It's the most precise tool we've ever built, but the delivery vehicle is currently too bulky for widespread, easy use inside the human body. On one side of the ledger, prime editing is, without question, the most precise and versatile tool we have ever engineered to manipulate the biological code. It possesses the theoretical capability to cure 89% of all known pathogenic genetic diseases. It accomplishes this without shattering the double helix and risking the catastrophic chromosomal translocations of traditional CRISPR.
It is a monumental leap in biological engineering.
But on the other side of the ledger, it remains a technology grappling with severe physical constraints. The delivery vehicle is currently far too bulky for widespread, efficient in vivo use across diverse organs. The software design requires painstaking manual laboratory optimization, and the permanent nature of the edits carries profound lifelong risk. It is a technology transitioning out of the theoretical perfection of its infancy and into its heavily scrutinized, highly complex adolescence.
The sheer velocity of the scientific progress moving from Andrew Anzalone's initial 2019 prototype paper to effectively curing patients of CGD in late 2025 is breathtaking. But the engineering challenges ahead are just as steep as the ones already conquered.
To recap the journey for you, we've moved from the blunt scissors and CRISPR to the precision word processor of prime editing. We've explored the Cas9-RT machinery, the elegant flat mechanics, the clinical triumph of PM359 for CGD, and the massive delivery challenges that lie ahead.
It requires a synthesis of virology, bacteriology, and endogenous human cellular mechanics to execute a single, perfect edit.
I want to leave you with a lingering thought, not explicitly debated today, but building on the sources. Prime editing's ability to safely alter the genome without breaking DNA brings us perilously close to routine germline editing.
Altering the genetics of human embryos.
Exactly. The barrier to germline editing has historically been a barrier of safety. You cannot risk shattering an embryo's chromosome with traditional CRISPR. But if we possess a true word processor for human DNA and we solve the delivery bottleneck, at what point does correcting a disease blur into actively writing the genetic traits of future generations? Where is the line between therapeutic editing and human authoring?
We're rapidly transitioning from merely reading our biology to actively possessing the tools to write our evolutionary destiny.
Something profound to mull over. Keep questioning and keep exploring. Thanks for joining us on this Deep Dive.